Good morning, everybody. Wim de Koning here at the Lincoln University campus, BioLink Innovation, and we will start a really exciting webinar called The Rise of Bioengineering. Before we're actually going to do that, and before I'm actually going to introduce our three absolutely fantastic speakers, a little bit of housekeeping. Webinar will be recorded, and we will actually mail it out to you as well, so that you actually have a link, so that you can actually see you can see it again. On the top of your ribbon, there is a Q&A button. Feel free to pose a question to any of our speakers or to all three speakers at the same time. And at the end of the webinar, so when all three speakers have given their presentation, we'll do a Q&A session. All participants are muted, and so feel free to get yourself a cup of coffee because yeah, we will not get jealous because we won't be able to hear your coffee machine grinding these nice fresh coffee beans. In the meantime, for the people that have no idea who BioLink Innovation is a full business unit of Lincoln University, and basically what we do is we bring people together. And very much like we're doing currently in a virtual environment, where we bring people together so that they can actually connect. And we are absolutely convinced that if we bring people together and they are like-minded, or they share the same passion, and they actually start to talk, and those conversations most of the time will lead to collaborations. And we truly believe that if multiple people actually start to collaborate, then true innovation actually takes place. Like I said, we're a full business unit of Lincoln University. I will introduce the first speaker, and I'm really excited that we have a really fantastic lineup of speakers this morning. First speaker is Professor Emily Parker from Victoria University of Wellington. She got her biological chemistry degree at Cambridge University, and she is the principal investigator and on the management committee of the Maurice Wilkins Center for Molecular Biodiscovery, which harnesses and links New Zealand's outstanding expertise in biomedical research to develop cutting-edge drug and vaccines, tools for early diagnosis and prevention. She also teaches at Victoria University of Wellington. So lucky students out there to have Emily as their lecturer. So without any further ado, I will stop sharing my screen, and I invite Emily to share her screen. Thank you so much.
Thank you, Wim. I'll just see if I can make this work. I hope that all works for you and you can hear me clearly.
Perfect.
Thanks very much, Wim. I was just confessing to Katie and Wim that I'm not sure about this billing as an excellent contributor, but I am very much interested in bioengineering in New Zealand. I'm very keen to see this field develop further from where it's been. And I also confess to reading the brief of what I was supposed to talk about late last night. So, you know, you've got my ramblings and my thoughts and some other perspectives that I hope are of interest to you. And of course, I'm really interested to hear, you know, comments that come through from people who are listening as well. I am, as Wim said, I am at Victoria University of Wellington, where I'm part of the Ferrier Research Institute. My team doesn't operate out of Gracefield; it operates out of Kelburn campus. My team is synthetic and chemical biology there. For about the last year and a bit, I've taken a half-time secondment to the Ministry of Business, Innovation and Employment, where I'm science advisor there. That's been an interesting time, particularly interesting for someone in my field because of the gene technology reforms which have been going on as well. So I'll only talk a little bit about that, but that is also something which is very relevant to the opportunities that there are in bioengineering for New Zealand coming up. Okay, so just my work and what I'm interested in, to give you a little bit of my scientific background. This is a slide that actually one of my PhD students made many years ago now, which kind of indicates some of the things that I like to play with. I really like biological machinery. I really like the wonder of how molecules, gene encoded molecules, can do exciting things. I was trained as a chemist. Now so much of my work uses the tools of biology and the power of biology, but the power of biology really to do some quite exquisite and complex chemistry. So the work that my team is involved in is about, sometimes some of us around enzyme structure and function, thinking carefully about some of the bond breaking and bond making steps in enzymes, how enzymes have evolved, and how they move and how they respond to molecules, how they sense molecules and change their activity, and proteins respond to their environment. But more recently, and perhaps the work that is most relevant to the topic of this webinar, is our work in the fungal factories area, so call it the work in bioengineering or biomanufacturing, where we've been looking at making using biological componentry to make really complex molecules which may have interesting uses. So as I said, I read the brief late yesterday afternoon and realised that we were talking about bioengineering, and just quickly looked up a few of the definitions that I had at my disposal when you look up how bioengineering is defined. And we've got some of our universities have been very have given offerings, degree offerings in this field, and some very strong work has been done in the bioengineering space. You'll be aware of the Auckland Advanced Bioengineering Institute, and their definition here of bioengineering was the application of engineering and technical innovation to human physiology and medicine. You've got a similar one just above there from the University of Otago, with engineering principles to address the challenges in the field of biology and medicine. So a little bit broader. And the bioengineering at the top, this is the Canterbury University one, involved in the application of biological and engineering to principles to the areas of medicine and healthcare. So a lot of medical stuff and a lot of different stuff is the way that we perhaps characterise bioengineering in New Zealand. I guess my perspective and my work, puts a broader definition on bioengineering, where I think it really is this way of thinking about how we manufacture and control and utilise that power of biology for a whole lot of purposes, not just in medicine, but to improve disease prevention, treatment, agricultural production, energy sustainability, and far more. So that complex way in which we can put the biological componentry together to make useful systems and understand our living world. So like I've just been saying, a lot of bioengineering, that broad topic of bioengineering can be around mechanical parts, changing things, as some of the images on the left-hand side of the slide do. Some of the fascinating computational skills that are happening, some of the different AI stuff that is happening in the field of medicine, all falls under that broad definition of bioengineering. Whereas I guess my focus and my attention has largely been on the right-hand side of this slide, where I'm interested in how we actually looking at the gene-encoded biological componentry and how we might be able to put that to work, how we might be able to understand that componentry, put that to work, and use that power of biological systems to feed and source fine chemicals, to do a whole lot of applications which are useful for us and our place in the world, and to protect and to enhance our environments as well. Okay, so I guess, especially in the field, and much of our fields has been much change. But I guess if we look at the last couple of decades or so in the field of bioengineering, particularly where it relates to DNA encoded machinery, what has really changed? And there has been a lot of change. Of course, molecular biology tools and ability to and understanding that DNA did encode really the biomolecules that give rise to life has been known for a long time now. For well over 50 years, those tools have been fairly mainstream. They've got better and better, of course. But what's changed massively? And if I think about it, I think, in my world and what I do, that availability of sequence data, the fact that we've got so much sequence data at our fingertips is enormously powerful. That, I mean, I've often say to my team and things that my dream in life is to be able to read chemistry from sequence. We have such a wealth of data out there to be able to understand what that data tells us, what that says, how we can use it, how we can put it to use, how we can understand it, how we can respect it, and all of that it comes with having that amount of data. And of course, what we've now seen is really quite precise methods of altering DNA, very targeted methods, sequence-specific methods, enabling us to do precise changes, introduce precise new genes or modifications. So that ability to both read and write genetic sequence has changed. When I first started working on enzymes, when I came back from the UK to New Zealand and first started wanting to work on enzymes, that was actually the time when I learnt the most molecular biology, because I was determined that I would be able to play with big molecules and small molecules alongside each other. So I was really keen to be able to do that. Of course, we were cloning up and PCRing up everything from genomic DNA and needing the source organism, of course. Now, of course, we access synthetic DNA, and that has really changed. The huge other thing I was going to mention is the change in understanding and use of RNA as well as DNA. Just wanted to make a point about the integration of data. I mentioned the rise of AI, and if you look at much more commentary around the fields of synthetic biology, you will see, and I'll talk about that in a moment, you'll see we're really talking about that convergence of techniques, those boundaries that were between disciplines, between computational science and biological science and chemical science. I believe that those boundaries are far more blurring, and we need to think differently about the way we undertake our research and we look for innovation, because those boundaries are not boundaries that have to constrain us. We need to find ways to be able to traverse those boundaries in ways which help us to create innovative solutions. And as I said there, that convergence of techniques across the life science, across the chemical science, across the computational science, it really challenges those discipline boundaries, the way we think about our science and the opportunities that there are. I said briefly that I had been involved in the gene technology reforms as part of my role as Department of Science Advisor here at MB. Many of you will have seen the announcement the government made in August that gene technology reforms to regulatory reforms are on the way. My team thought this was quite funny how I got the plastic lab coat in this picture, whereas our Prime Minister and Minister got the real lab coats, perhaps the real scientists there. So the other thing that I've been involved in during my time at MB is this OECD panel, GFT Tech Synthetic Biology. This has been quite fun, apart from the fact that it meets in the middle of the night, of course, because it's mainly suiting timeframes quite outside our own. But it's been fun because it's so expansive in the thinking in this area, in terms of the opportunities, and with a whole range of considerations. I mean, obviously thinking about the areas of health innovation and resilience in health, the health implications of synthetic biology, or their ability to utilize biological componentry and put it together in defined ways, engineer that biological componentry, food security and soil regeneration, issues around net zero and circular carbon as well. Of course, they also have talked OECD thinking about that world issues, thinking about distributed manufacturing, sources, food stock sources in different parts of the world, and being able to manufacture closer to where products are utilized. And of course, that, as I mentioned before, that AI and synthetic biology, that convergence of data. So there are a whole lot of considerations: equity and access, biosecurity and biosafety, of course, thinking about responsible innovation, how that is done responsibly, and especially with the technology advancements that there are. Developing a skilled workforce, something I'm really passionate about, making sure that we have in place in New Zealand, that we set ourselves up and our economy up for a successful future by developing people and building strong, resilient innovation and ecosystems. Okay, just to put some perspective on this, and that, where things are happening globally, there's been a lot of international interest in the biotechnology space, particularly in biomanufacturing, where there has been quite large projections of value and some large investments made in other parts of the world. That ability to use the power of biology in the manufacturing process does offer in many ways an attractive alternative to other forms of production systems. As it says here, to make things like plastics, fuels, materials, medicines, a lot of opportunity in there. There's a lot of hype. There's a lot of potential. The things I've been thinking about is what part New Zealand can take in these changes, and what are the opportunities for New Zealand as we go forward too. There's been a range of different reports, and like I said, thinking about that bio revolution as synthetic biology has come on board. Lots of claims of disruption, lots of ambition. We're only starting to see some of that stuff come through. So here in New Zealand, what am I seeing? And this is just some personal perspectives. There's a lot of noise, and I'm sure Zara will talk about Biotech NZ's appetite for advancing the science in this space, advancing the opportunities in this space. And there has been strong appetite for regulatory change, which was why the government has responded in that way. We are seeing some exciting startup activities which are happening. We're seeing a growing awareness of developing IP in this place, and that is good to see those opportunities coming online. And we have, in my opinion, a strong need for scale and joining up the dots. We need the successes, and we need to think carefully about our place, our ability to do it. We also have very limited biomanufacturing capability at the moment. This is growing, and there is ambition. There are a range of different people setting up opportunities in the biomanufacturing space, and there's also regional interest in biomanufacturing as a way of developing industrial capability in the regions as well. So what I want to say here in New Zealand, I guess from my perspective, we need to think carefully about our distinctive opportunities. Think about what place we play, what are the opportunities we can capitalise on, what's distinctive about being in Aotearoa New Zealand, and what we can use, what are our local sources of feedstock, what are our unique opportunities. I think we should chase activities that lead to a spectrum of activities. Well, I decided this at the last minute. So I'm very keen that we create jobs in New Zealand. We generate industry and activity in New Zealand. We need to think about our export markets. This is not just about supplying to our local markets. This is about giving us far more international market. And we need to think carefully about scale, about market size, what we can take on, what we can handle, and how we upskill and keep as much value associated with our innovations in New Zealand. So just go quickly and tell you a little bit more about our research in this area. I mean, my research is very much interested in this area, in the power of fungi to be very, very clever chemists, in that they can make exquisitely powerful bioactive compounds, and they can make them do this complex chemistry that we find much harder to do in the lab. So, as I say, fungi are clever chemists. We have made use of their ability to make important bioactive compounds for many, many years. And I've just got a few examples down here of compounds that we have put to use because of the ability for fungi to do them. Of course, most fungi don't do that because they think that we want them to do that. They do that because they need to make those compounds for their sustenance and their biological needs. But we, of course, can make use of this compound, make use of these compounds, make use of the ability to biosynthesize and put this to use. Now, of course, the fact that they can make these compounds means that they have the gene-encoded machinery to be able to make these compounds. And so what we have been doing is looking at that componentry and putting it together in different ways so that we can make both new compounds but also upscale compound production where there is useful applications of those compounds. Class of compounds that we have been working on is these compounds I call sorry about the chemistry for people who are not chemists. But aren't these compounds wonderful? I hope you're enjoying them in all of their glory. These compounds called the indole diterpenes, which are amazingly complex group of compounds, very, very difficult to manufacture by traditional means of chemical synthesis, but they have some powerful bioactivity that we want to be able to access. So what we have done is put to work what we have done is chased a compound known as notosporic acid, which is a potent insecticide. This is made in a fungus which is endophytic, that lives inside a plant, that flower there, Bontia on the left-hand side. So what we have done is gone in and take out those genes, find the componentry, the genetic componentry which was responsible for making this compound, taken it out and put it into another more pliable fungus, and we have generated a biomanufacturing system through heterologous biosynthesis. So we put those genes to work in a way that we can upscale the biosynthesis of this really potent insecticidal compound. Very, very complex chemistry, just to scare you all. It's been a lot of fun working this out and working out how we resolve and make an efficient biomanufacturing system. So that's what we've been doing as part of that. I'm going to finish now. I'm going to finish and just to thank all of those people who are involved, my team, all of those people who have supported me, and of course, very happy at the end of this time to take any of your questions or comments or your own musings about where you think things are going to in this area. Many thanks. I'll stop sharing if I can.
Emily, thank you very much for that insight. And I already have a bunch of questions, so we're going to have dig in a little bit more in what you said. For the people that are watching this as a recording, we do apologize that you have not been able to access the live webinar, but we still hope that you enjoy the recording. Without any further ado, I would like to introduce our second speaker of this morning, Sarah Champion. Sarah Champion is the Executive Director of Biotech New Zealand, and she has an impressive track record with over 20 years experience in commercial and senior roles, looked at strategic development, biotech and health, and at the food and beverage sectors. Like I said, currently she is the Executive Director of Biotech New Zealand, and she's leading the team that is maximizing bioscience and technology capability to create a strong New Zealand bioeconomy. Sarah, I will mute myself and the floor is yours.
Thank you for that. I'll just share my presentation.
Absolutely perfect. We can see that, Sarah. Thank you.
Excellent. Kia ora everyone, and usually I say nice to see you all, but actually today you're all just symbols. So again, really nice to see a number of those symbols on the screen. Hey, I've been really fortunate to come back from 2 separate trips overseas. I first attended the Crop Life Summit. So they are an industry body, again, looking after the kind of very much focusing on the new gene regulations in the space in Australia. I went over to Canberra, and again, as I said, right now Australia is looking at updating their regulations as well. I have also just got back from Oz Biotech conference in Melbourne, and what I did see there is a significant amount of investment going into the infrastructure around certainly the human health biomanufacturing. The reason I'm highlighting that is because I've had a lot of opportunity to catch something on a plane. And right now, as you can hear, my voice is pretty laboured because I've managed to get some international bug. And right now, very much one of those biomanufacturing facilities. And now I just have to make sure I don't scale up and spread what I have. So we'll keep this a little shorter because my voice is pretty stretched. However, today I was asked to give some of my insights, kind of where New Zealand is currently with our regulations or our legislations around bioengineering. I am probably going to have a little bit more of a focus on that kind of the food, agricultural space, environmental, industrial, because there are significant differences when you start looking at the human health bioengineering space. I was also asked to sort of talk a little bit about sort of some of those consumer demands or those consumer insights when we are developing our products, certainly in that bioengineering space. What is some of the support available or advice for companies that are looking at using this technology or certainly facing some of those challenges around this technology? I always love this, what does the future look like? So again, a little bit of my view there and highlighting the work that we do in Biotech NZ. Firstly, just like Emily, I did that whole quickly looked up what actually the definition of bioengineering is. And you can see it's quite broad depending on kind of which area you're looking at. I really have now grouped both kind of biotechnology and bioengineering a little bit together. So I'll kind of interlink and use those terms because kind of for me, we both need each other, right? So biotech is about manufacturing those biological systems, and bioengineering is what is required to manufacture and scale up certainly of those products. Before I jump into where New Zealand is with the regulations around biotechnology, I want to say that New Zealand does the traditional biotech incredibly well, and this needs to be noted. So certainly up here, when we look in the agricultural sector, we do single selection, we do crossbreeding, we got mutagenesis in our tissue culture. We do this all under the umbrella of the non-GMO banner. And I really want to highlight that our companies punch above our weight, both across the world with our farming, our livestock, and our horticultural products. We'll probably hear, you know a lot of these companies, but of course LIC, certainly with their cattle genetics, plant and food, and their relationship with Zespri around the gold, the red kiwi fruit. But when we start thinking about the bioengineering, we start thinking more around some of those closed systems, the fermentation, the bioreactors, and those controlled environments where we are using those microbes, we're manipulating them to do the things that we want. And again, we still do some really amazing things. Our companies who are again doing using these techniques without genetically modifying their microbes. And a lot of this area, or these companies are in the area of kind of converting waste streams into kind of high value products. I think we've all heard of mint innovation. Again, extracting those kind of critical metals, gold, for an example, from the e-waste. We've also got cetogenics, which again is recovering those kind of carbon resources. And of course, I mean, when I think of waste streams, I always think of the great work that Scion does, certainly with their products around the bioplastics, the biofuels. But really where we are right now in New Zealand is the big hot topic for New Zealand, which is we are not able to use these modern biotechnology tools. And Emily's highlighted a number of them. And again, certainly those sort of new genomic technologies, you know, new breeding techniques, precision breeding, genome editing, and of course, gene editing. I just put this slide up here because just like any other country in New Zealand, we have a highly complex regulatory framework, which is all kind of interlinked. I've taken this from the Aotearoa Circle report. It's a great document if you haven't read it. I just want to highlight that the HASNO is really our primary legislation when we're governing applications of genetic technology. The HASNO sets out a really comprehensive regulatory framework for assessing, managing, and controlling the risks associated with genetically modified organisms. The Act applies to a range of activities, including the imports, certainly the manufacture, and of course those releases of those GMOs into the environment. Also in there is the EPA, the Environmental Protection Authority. This was established around the same time as the HASNO Act. And this is a body that is responsible for overseeing, again, the importation, the development, the field trials, and the release of GMOs. I want to also note that when we talk about genetically modified food, it sits under a different regulatory framework and it's regulated by the Food Safety Australia and New Zealand. We call it FSANZ. And is not, again, subject to that regulation under the HASNO Act, which is, again, important. If we start looking at our HASNO Act, the first thing you will see is 1996. I always think about what I was doing in 1996, and back then I didn't even have my first cell phone. So that just highlights to you how old this Act is. And again, there is a lot has changed in the world in terms of technology and how far this has moved. In 2021, there was the Productivity Commission report, and they basically said the government should review the GM regulatory framework to ensure that it is fit for purpose because of these huge significant changes in technology. And of course, gene technologies or gene editing is offering a new huge amount of new potential opportunities for, again, boosting productivity, improving health outcomes, reducing biosecurity risks, and responding to some of these climate change risks and other environmental problems, both effectively and efficiently. I do really want to iterate, though, that New Zealand does not commercially grow any genetically modified crops. That's including anything that is genetically gene edited, and we have no fresh produce or meat in New Zealand for sale. And I also want to highlight we don't have any commercial closed system products in New Zealand that we're commercially manufacturing. So what does this mean for our company or our product right now? How important is our consumers when we are developing these products? And the first thing I do go back to, and it's pretty exciting to have Emily on the call, is she is driving the regulatory change or regulatory updates, and again knows that our ecosystem is really asking for these regulations to be updated. We need to have access to all the tools in the toolbox to for some of these big challenges that we're facing. It's really interesting when I talk to a lot of people about biotechnology, bioengineering, as most people or most companies don't actually fear the technology. They don't just know really how to engage with the technology. So they're asking me, we have these products, how do we engage with this technology? And the first thing I'm saying is, let's just take a step back and actually ask, what do your consumers want? What are they asking from you? What are they looking for? So are they looking for different types of varieties? Are they looking for price difference or lower prices? Are they concerned about their health or wanting solutions for some health concerns or have health benefits? Or is it around kind of that environment and impact of a product or your product? So what I'm wanting people to really start thinking about is not the technology, but actually kind of your end product and what your consumer's asking. So it may be around what the product is, but you may be taking a step back and thinking about, well, actually, it's not our own product. It's actually the way the product's made, or we want to future-proof our product to ensure that we are still in the market. Future-proofing your farm is really important. And when I talk about farms, I'm talking right through from kind of that horticultural right through to, beef and lamb. Now, I do speak a lot at the moment to companies who are actually using a lot of the our kind of current farming systems. So again, how do we increase our yield on our farm? How do we reduce the pesticides? How do we add nutritional benefits? And also, again, this climate resistance. But I do know today we're talking a lot about kind of those disruptive technologies or solutions to those traditional farming methods, or adding value to those products that we always have. So I think it's really important, again, to start thinking about who your consumer is, because we have seen a lot of this kind of boom and bust when it comes to biomanufacturing or kind of the synthetic foods that have been produced. And so, again, there's very different views when you're talking to consumers about health or vaccine developments or anything regarding kind of health in the medical space. You'll also have very different conversations with your consumers when you're talking about converting waste streams, and you'll have those very different conversations when it comes to food. And that is probably at times the hardest to get across. And we're seeing that both when we're talking about using modern gene technologies. Again, human health, there's a real comfort around that, right through when it comes to in your food. When it's been genetically modified, we see a very different consumer reaction. And again, you will see this within that kind of synthetic bioengineering space as well. I think one of the things that I do say is that New Zealand's not an early adopter of this technology. We're not a fast follower. Therefore, we do have a significant amount of data that we can look globally to help make those decisions. And that's really key when you are thinking about engaging with this technology or looking at new product development or future-proofing your current product. There is a lot of support out there. And I think reaching out to your industry bodies, there's a number of industry bodies that sit in this space. I also really want to highlight some of the government agencies as well. So NZTE do a lot around that understanding your consumer, your market. I would be reaching out to them. I really want to highlight Callaghan Innovation. A lot of the times we think about Callaghan as connecting people, connecting them to researchers, but also grants. But I also want to highlight that they do have manufacturing facilities, certainly in that fermentation and bioprocessing. They will help you in terms of your R&D. They will do small batches. They look at some of the scale up, but they're also got a lot of expertise when it comes to, you know, the food and beverage sector, live cultures, in the precision fermentation, whether it comes to, you know, enzymes and small molecules, and of course the human and animal space, plus of course biocontrol. So I just wanted to make that known and aware if you're not sure. We've also got BioSouth. They're in a high growth curve, again in Christchurch. They are a contract manufacturing for, again, those bioproducts in that kind of microbial fermentation space. They have, again, help and support in scaling, plant design, operations, and are really committed to helping the ecosystem. I would be reaching out to them for a conversation. I'm really impressed, again, with the Crown Research Institutes. It's been a tough time in New Zealand where we aren't allowed to use modern gene technologies, and they have held the capability, the staff, the people, the knowledge, and the research, which will actually take New Zealand forward significantly once these regulations come in place. So again, another great resource to be reaching out for them. And then, of course, those partnerships, whether they are international and national. And one of the things I see when I was in Australia, we can't build those manufacturing sites to the scale that Australia has. We don't have that kind of money. We don't have that kind of pipeline. However, a huge opportunity here to be thinking about who those partners are just across the ditch in terms of with Australia. People ask me around what does the future look like? Well, just let's have a look at what now looks like. And I just wanted to show you, certainly in New Zealand on the left-hand diagram, everything right now and all those modern technologies, gene editing is all under the GMO. It's all red. Also, when you look at a global map, again, you know, New Zealand is one of the few left in the world that has red. So that's us now. We are moving forward, and it's a very, very exciting time. What the future looks like, I have just put up a little bit of a chaotic diagram, and I'm loving ChatGPT. So I was asking it last night to draw me a convergence of chemistry, physics, biology, and tech, because I think that's where our future state is. It is not where we start talking about green tech, blue tech, biotech. We're actually moved forward, and we're actually thinking more about those interdisciplinary thinking. We're actually starting to think about what tool do we need in the tool belt to use. Oh, I need a little bit of biology and I'm going to add a little bit of tech. oh, that might actually be using some of those big data systems right now sitting in the quantum tech space. How can we utilize that brain power of quantum tech when it comes to our drug discovery? So my big future state will be thinking about, as I said, stop thinking about these silos. We're starting to think more about this interdisciplinary thinking. We think about the solution. What is the solution, or what are the challenges? What are the solutions, and what are the tools we're going to use? And it won't be, I need a biotech tool. It will be a multitude of tools to come up with a product. And again, I do think that consumer engagement is absolutely key. So again, think about the solution, think about what your consumer wants, what is your consumer asking for. You will again see those, a real sustainability focus coming into your products, and therefore we are going to be looking at the synthetic biology, manufacturing our food in a potentially in a cleaner, easier, faster scale-up way. We will need to have a more flexible regulatory environment. That's a real challenge while we're currently looking at updating the regulations. How do we future-proof this, especially when technology is moving this fast? So again, future state really is this kind of blend of interdisciplinary science. Right at the beginning, when I was introduced, we talked about Biotech NZ, and I did want to just highlight we are an industry body. We have absolutely changed the landscape in New Zealand. I've been in my role for 7 years. When I first came into this role, there was nowhere that I saw the word biotech in any of the government documents. I was at the point where I was thinking of changing the name of Biotech New Zealand into something that doesn't even have biotech in the name, because it was it felt like it was such a dirty word for New Zealand, certainly where we've come from with the kind of the anti-GMO. We stuck to our guns. We've worked incredibly hard across certainly the ecosystem, pulling the ecosystem together, working with government, working with the government agencies. We now have a very close relationship with both the National Party, but also MBIE writing these regulations, and we're there to support in any possible way. What I've put up here, if you don't know, that in Biotech NZ is actually part of the Tech Alliance. It's a real force. There is a lot of tech in there, and so when it comes to biology, certainly Biotech NZ and Agritech a little bit in that biology space. I'd love to see a lot more of that chemistry and physics coming through, and of course quantum is another area of excitement. But I do want to show that we're part of a credible wheel. We have certainly, as a collective, we've been very impactful, and we, as I said, have worked very hard. And I'm very, very proud of what we've achieved. And thank you very much for giving me the opportunity to kind of highlight some of my very high-level views of where I think this tech's going to take us in New Zealand. And just want to say a very, very exciting time, a really exciting time. So again, thank you, Emily, for the work that you have been doing in this space.
Sarah, thank you so much and for your contribution, and absolutely fantastic. I will introduce the, I can actually show that. I will introduce Dr. Richard Scott from Ag Research as our last and final speaker, and then we'll open the floor up to some questions. Richard has a PhD in plant biotechnology, and he leads the climate change and forage innovation team at Ag Research. He also leads the Gene Technologies and Enabling Platform, and which was established to build strategies around the application of gene technologies. So without any further ado, Richard, the floor is all yours. Yeah, and I can see your slides. I can't hear you.
How's that?
That is absolutely perfect. Thank you so much.
Right. Morning all. So I started in plant biotechnology probably back in the late 80s. So it's been close to 40 years that we've been using this tech in the forage space or in the agricultural space in New Zealand. And it is now that we are seeing an opportunity to actually deliver something on farm that's going to be for the farmers rather than and for the if you like, consumers and the environment. So I'll start off with the story behind HME ryegrass, and the concept was to improve the energy content of ryegrass, which is the main forage used on our farms across the country. So the problem was around nutrition. The constraints on animal production was primarily due to the feed available, the quality of the feed, and not the animal genetics. And just to give you an idea of that, in New Zealand, dairy cows, 4,300 litres per cow, and in the USA it was effectively double that, but the difference there was actually in the type of feed they got. Also, it was widely considered that the ratio of protein to energy was too much protein sort of thing. So we were going to look at one of the part of that problem was actually rebalancing that ratio. So the solution that we saw, and this was over 20 years ago now, that this project started, was that we'd use oil to change that energy balance. And gram for gram, oils were twice the energy of carbohydrates, protein, and that foragers have very little oil compared to the feedlot industry. The seasonal variation in leaf oil content is far greater than the genetic variation. And what this meant was that actually we couldn't use genetics to get that level of variation that was needed, that large scale, if you like, that shift in oil. And the other thing was that if we did make the oil, then oil is typically phenotypically neutral. In other words, it wouldn't change the morphology of the plant. If you look at sugars and that sort of thing, they do change the, if you like, the osmotic potential of plants, and you end up with all sorts of other effects. So the concept was to genetically modify ryegrass to increase that oil content. So there were already many genes known for lipid biosynthesis or oil biosynthesis, and also plants, they make high density oil bodies for long-term storage. So we were going to use two genes. One, increase the oil, and the second one, to wrap it up and protect it from saturation. And now, why saturation? Because in the rumen, fatty acids soak up hydrogen produced by the, and this is part of the reason why methane is produced. It soaks up extra hydrogen ions, and fats can become saturated. So our idea was take unsaturated plant fats, put them into, allow the animal to eat them. They don't get converted to saturated fats, and you have a healthier fat in that final product. So the technology, first gene was DGAT, which is diacylglycerol acetyl transferase, and it's the last step in oil biosynthesis. And the second gene to be used was oleosin, and that basically wraps up and encapsulates oil droplets. And it's the oleosin that probably was the main twist, or the main thing that we manipulated here. Oleosin has a hydrophilic component to it. We call it the hydrophilic arms, and a hydrophobic core. And what it does is basically all these oleosins, they wrap themselves around an oil droplet to produce an oil body. And what it does is it effectively creates a barrier that stops oil bodies from coalescing and forming large oil globules, or an oil pool. And that would be it wouldn't be good for manipulating by the plant, and the other thing is that it provides a smaller surface to volume ratio, so that the plant can access it rapidly particularly when the seed is germinating. So our first concept was polyoleosin. Why polyoleosin? Well, what we were doing was we were looking to increase that stability so that the oil would pass through the rumen, and so that it wouldn't be biohydrogenated and into the secondary stomachs and intestine. It would be taken up as an unsaturated fat. We had our polyoleosin, and what we were looking at is we were going to make a chain of them, try and increase the stability that way. So this was a genetic approach, a molecular biology approach. What we were going to use is we were going to use a chain of oleosin genes and to produce a chain of oleosin proteins. Now, our first iteration was to simply clone the same genetic sequence one after another, up to 5 times. And what we found is that we didn't get any increase in oil. And when we looked at the RNA, we found that it was highly unstable. And around this time, it came out that you get effectively silencing if you get too many copies of exactly the same sequence. The second iteration was actually designed on a whiteboard, and took about a month, and coming back to it and say, oh, well, we don't want anything with this sequence, and we want something here, and we'll scrub that one out. Yeah, it really was engineered, and then we emailed the sequence off to a company, and they chemically synthesized it, and sent us back a DNA strand. So that was, a major step for our system. And the key thing about this was we had exactly the same concept with a chain of oleosin genes, but what we could do is we could make the genetic sequence very different, but in each one of those components, but still end up with the same amino acid sequence. So we then avoided that problem with the RNA instability. And it worked. So we ended up with, this was done on the microbial scale to start off with. We were producing these chains of oleosin, and then we used them to basically produce artificial oil bodies. And we found out that, yes, we could encapsulate more oil, and that it was highly stable as well, so temperature stable. So that looked good. We then put it into plants, and we noticed that in plants, the oil would peak in young leaves and then drop away. And we couldn't figure out what was happening there. And what we thought was that actually with these polyoleosis, they're a chain, and what was potentially happening was that they were arranging themselves around the oil body. But what you would get is they were forming islands. So they weren't fully encapsulating it. They weren't properly integrated. You're getting bends and kinks in them and that sort of thing, which were points of weaknesses. And it was actually the plant was metabolizing the oil when it needed to grow. So it would come back in, it would store it away when it was young, but then when it started to really take off, it would come back and basically steal that oil back again. So the next step was to go from polyoleosin to cysteine oleosin. So if you consider polyoleosin as a three-dimensional molecule, you have multiple of them around the oil body, and they're, as it's being formed, they're in all different formations, and basically when it's formed, they're out of sync with each other. Now we had a problem. We had a problem with another project that we had ongoing, and that was there's amino acids that are called cysteines, and they cross-link, and they very tightly. And we had proteins that we were trying to look at, but we couldn't break these cysteine bonds. So they just kept on interfering with our analyses. So what we decided to do was use that as our solution for the HME project, and actually use cysteines to cross-link the oleosins, rather than putting a chain in. What we'd do is we'd put add a cysteine amino acid, and we'd let them do their own cross-linking. The idea being is that as it's forming, they're in different formations. It stops, you get cysteines next to each other, and they create these disulfide bonds, and then you get chains. You get chains of 2 there, you might get chains of 3, or you might get more. Back to the original oleosin. Take that, okay, polyoleosin, that was a head to tail chain. What we're looking here is we're looking to have cysteines, and we had to look at different combinations to figure out actually what was going to be the best, because this was blue sky, if you like. We didn't know anything about what it might look like. This was so we made a series of them with different numbers of cysteines, and again it's back to that. You have that outer layer, the triglycerol core, and then forming that shell, them cross-linking, and then forming that very strong shell around the oil body. Bit of tech in here. These are proteins, gels of different size proteins. So number 1 is actually smaller than number 2, if you're looking at those arrows there. And so for that band, those bands there, got 2 bands, and effectively what you've got is you've got 2 formations of your oleosins potentially. They're fairly just happen to align with each other. With these ones here, oleosin 1,1, what we've got is we've got a single cysteine on them, and they can link like that, or you get a larger protein if they've got 2 together, etc., etc. And then we get to the 3,3, and we've got all sorts of formations there, and that's why you get that blurred set of bands it was shown by that red circle there. Oh yeah, and just that the antibody was less sensitive, so that's why we had a variation there in terms of intensity. And when we looked at it, we found that the 3,3 cysteine oleosin actually led to an increase in fatty acids in the plant. So if you particularly around look at the graph in B, you will see that the wild type is in blue, and our 0,0 plant, which is a transgenic plant with oleosin in it, that was around that 35 milligrams per gram of dry weight, and our orange ones, those 3 plants there, were all higher in terms of leaf fatty acid. Basically no difference in morphology in that shown in E. And when we look at our fatty acids over the H on the right hand side there, a fatty acid over the age of the leaf, we see that compared to the wild type, we maintain that higher level of fatty acid as the leaf ages. And this is important. You can imagine making hay. Okay, your hay is still going to contain that higher energy content. So now we come to the HME ryegrass phase and switch. And what I mean by that is we switched from the focusing on the energy production and protecting the fatty acids from biohydrogenation. around 2011, 2012 sort of thing, the concept that fats could inhibit methane production started to gain traction. And so our, if you like, focus pivoted from that productivity to can we mitigate methane emissions. Our plants were in this range, around 35% sort of thing, 30 to 40%. That was their base level of fatty acids. What we were looking at is a increase that, if we could effectively double that, then we'd have a potential to give a 10 to 17% reduction in methane emissions. So these were theoretical numbers, and this was when we got, roughly in 2016, we got funding. So that was from MB, but we also had other commercial and systems, so we had dairy NZ in there as well. And PGG Wrightson's and Grass and Seeds, they were going to be effectively the distributors of the technology. So we had them on board to support the development of the program. So how did we get on? I won't go through the technology, I'll just come through to what we ended up. So we did put oil into ryegrass. Our HME ryegrass successfully developed it. We managed to increase the lipids by about 50%, from 4% to 6%. What did that do? Increased our energy content by about 10%, our gross energy, and that varied between roughly 0.5 and 1 megajoule per kilogram of dry matter. You talk to a farmer, if they can get a 10% more energy out of their feed, that's a huge difference to on-farm production and management. From that graph I showed before, an increase in that oil would lead to a 10% reduction in methane. We'd also end up with a reduction in urinary nitrogen, and that was because changing that nitrogen to or protein to energy balance. With that decrease, you get decrease in nitrogen loss into the soil and subsequent loss of nitrogen nitrification of waterways. We also found out that you get a 15% reduction in nitrous oxide. Now this was done by effectively pouring urine onto pot plant or pots containing HME ryegrass and then measuring the nitrous oxide outputs there. The other interesting thing is that with your increase in energy, you're going to get effectively your animals developing faster or eating less but developing faster, and that's also going to have an effect on your methane production. So potentially raising that up to above 15% reduction in methane. Something that's not often talked about though is what did we do wrong? What could have we done better? So in hindsight, fantastic thing. Actually, I've jumped out of scale here. So these issues generally occurred in the D phase, if you like, of the R&D project, of the program, but we did learn. So the organisational path was not aligned with the project path. We're a research organisation. Part of that measurements is based on papers, whereas with if you like, if you're in that development stage, you're probably after minimalism. You're pushing your product down that pipeline as fast as you can and aiming to get it out for commercialisation. Flip side of that is that part of that research, we got patents and it extended our protection of the technology. The other one was early engagement with the regulators. We got to the stage of where we wanted to go to field trials in Australia. And yeah, instead of computer says no, it was the regulator did. And this was around allergens. They had a particularly bad case of ryegrass pollen causing significant health issues during a storm in Australia. And they said, well, actually, you've got sesame in here as well. That's a significant allergen, and we don't want that being trialled or even released over here, so no point in trialling it. Now, we had considered this, but it wasn't a problem in the US. So we do have a parallel program in soybean going on in the US. It hadn't been considered an issue. But what had happened is during the development of the technology, the status of sesame as an allergen had changed, and it had become a significant allergen in the food, and listed as a significant allergen in the food industry. And the other one, the final one, was an unsuitable timescale for assessing assumptions. We'd made predictions early on. Early results led to calculations of timescales, but then we didn't monitor subsequent, how things were progressing. And so it's that project management, I'd say, would have been a key thing, that we just probably we had lots of balls in the air, and so that was pretty essential. I must have cut you out, Emily. So this is, recent announcements. We're seeing a change in New Zealand. That we originally started was, 20 years ago, with thinking, well, we're going to develop this technology so that New Zealand is going to be ready to go as a global competitor when things start changing globally. Things did change globally, New Zealand didn't. It looks like, fingers crossed, that we are going to play catch up. So I'm looking forward to that and seeing how these changes pan out. This was a field trial in the US, and what I'm hoping for is that in 4 or 5 years' time, we'll be able to easily have these in New Zealand. Now, that was 5 years ago. Just to jump on quickly from there, is that we have just completed a field trial and analysing the results at the moment, where we've done an animal feeding trial. We grew these plants, in the glass house. We weren't allowed to grow them outside. Massive cost, and took a long time. And so it's basically that first field trial, or the first animal feeding trial, of this product being fed to livestock in New Zealand, and we're probably about 3 to 4 weeks away from having those results back. So, like I say, the future is looking very exciting. So, thank you very much.
Thank you so much, Richard, for that. And may I ask the panelists to come online again? Switch on your. Thank you so much. Fantastic. For the attendees, please put some questions in the Q&A if you have them, and we will actually have a session. And but I'll start off by asking some questions so that the people online can actually think of better ones. Emily, may I start with you? And it's really good that you emphasize that bioengineering is more than just creating more medicines, and so and I think Sarah emphasized that as well. It brought me back to my time when I was doing my PhD, and I figured out that when I was sequencing that I had to send little snippets to a database in Heidelberg to actually get my analysis and my analysis done, and if there were any matches to my DNA sequence data. At the moment, you're basically saying it's cheap and there's lots out there. Sarah also showed something that artificial intelligence could actually create a little picture if we tell it to actually do that. How would you interpret the context of the availability of the DNA data that is lots and cheap, in potential being used/misused by artificial intelligence?
Yeah, I mean, I'm sure the others will be interested in commenting on this as well. I think there is a challenge with that data that is out there. In some senses, as I kind of alluded to, we don't yet have all of the skills to read that data that is available fully, though I am anticipating that AI tools will make a massive difference to that. And so when we sequence genomes and have sequences open access, that provides an enormous tool for innovation. It also means that it can be seen by some as a means of exploitation as well of that sequence data. And that's something which is really relevant to this country in terms of data sovereignty, and something that needs really careful thinking about. But certainly the emergence of the AI tools alongside the data that we have available to us, and part of my work that I alluded to very much into protein sequence and structure, I guess some of you may be aware of the advent of AlphaFold, and that really came about because of a huge database that has been over many decades has been assembled of sequence data and structural data of proteins. So that huge database has been assembled, and as AI tools came on board, and sophisticated AI tools much more readily now, we are being able to predict structure from sequence, which takes out some of the sometimes really complex work done in getting a direct structure and experimental structure of a protein. I suspect those similar innovations will come across the field, which are able to take that sequence data and to utilize it as well.
Thank you, Emily. Sarah, Richard, do you want to comment on this, or?
I'll just add that it's you talked about how long ago you had to send that information away to get it analysed, and in those early days that information that you're getting back may have had flaws in it, and that sort of thing, and it's taken, and you look at it now and the level of detail and accuracy and complexity in that information now. And I think the thing is that AI will follow that same path, but it's going to be much shorter. It's going to get it wrong in some places. In the early days, we're going to have to recognise that, and and just be aware that it might be in double check, where we can or wherever. But it's still going to be rapidly changing and be just light years ahead of where we are now, very fast.
I'll move on to another question. If you don't want to comment, Sarah, that's fine. Richard, you have created this genetically modified ryegrass, and you basically showed us that it's not magic and it's science, and that we're actually going to make procedural mistakes, and that it was a lot of trial and error. But that it is a really valuable tool in the toolbox to mitigate against climate change. Will things like artificial intelligence help you to speed that process up?
No, I wouldn't. I think the thing is that those concepts were still, came out of the human mind. They may speed it up in terms of you'd be able to say, right, for that sequence, I want this taken out, and like I was talking about on the whiteboard, how we designed it on the whiteboard. But in terms of the actual hands-on work and getting it out into, still getting it out into pasture, that's still the farmer. There's no particular room for AI there. Minor impact on speed and delivery, but other than that, not really, I would say, for AI.
Okay, thank you. Sarah, one of the questions that you asked yourself and us, what do the consumers actually want? And you alert to the dilemma between product and the production methods. You said that probably a social acceptance in medicine and waste is probably there, but in food it is still a question mark. How do we stay competitive and protect our New Zealand consumers at the same time?
And, sorry, in terms of how do you mean by protecting our consumers?
Well, I think that if we're looking at precision fermentation, we perhaps start producing novel proteins using genetically modified technologies. If, of course, that will be allowed here in New Zealand. How does we could produce a food that the consumer really wants, but how then we align that food with our production technologies? Because, like, I've seen some beautiful slides from Emily using and showing us the chemistry, but I would assume that the average consumer in New Zealand will be completely out of their depth.
And I think just kind of the first number one, that we are still working within a regulatory framework. So when we update our Has No Act, it is a framework for, again, safety and risk. And so it's not all, all of a sudden we kind of tear down all of the safety mechanisms and we're just free for all. There will be very much different methodologies will sit in the different risk categories, and that will be assessed, as I said, by the regulatory framework. But also there'll be a compliance component as well that sits within that. So again, that good governance, we see it globally. We talk a lot about stewardship, which means that you are doing the best, you are producing a product that you say it is. If you have got that regulatory okay, you are actually producing that product to your consumer. So when you sort of talk about the safety of our New Zealanders, we are in a framework. Now, when it comes to what types of products our consumers want, it's really going to be, there's kind of two things here. We're going to have that import market that's just going to come in. And so people will have a decision on what they want to buy or what they want to eat or what they want to consume. We will see as soon as those the regulations change, like the rest of New Zealand, whether you talk about medical devices, pharmaceuticals, it's all about 95% imported. When we start looking at what are the types of products New Zealand companies and New Zealanders will be looking at producing, again, they'll be looking at both that international and our internal market as well. I really hope there's a lot, we continue to do a lot of work to support our New Zealand ecosystem, our New Zealand fauna, flora, and certainly, helping some of our New Zealand challenges, which are slightly different, say, than the rest of the world, just due to our farming system. So I think, we have a regulatory framework, and that's kind of my starting point.
Fantastic, Sarah. Thank you so much for making that really clear to everybody. We have a question from Rika online. Thank you so much for the presentation. I'm curious to hear your viewpoint on developing alternative proteins in New Zealand in the near future. How is the development of these alternatives seen in your work?
I'm just going to jump in here. And we have the likes of Fonterra, who are not doing this research here in New Zealand, but they are looking at, and they have companies that they've invested in this space. So when we talk about viewpoints and alternative proteins, I do believe this is a huge area of opportunity, and it certainly can run in parallel with our current farming systems. But again, it's just another alternative way of developing, different products. So that's, we look at kind of what our big companies are doing.
Clear. Thank you so much. I think I will have a last question, and I would love the three of you to actually give us some feedback on that. Sarah, you said that, or at least that's what I interpreted, so please correct me if I'm wrong. New Zealand is very much a leader in the traditional biotech space with our traditional breeding and enhancements of agriculture. We also are creating now this new framework around how we're actually going to use genes and into the future within the New Zealand context. I also saw a graph with a lot of different acts that are closely aligned. In a perfect world, and I realize that it doesn't exist, but let's assume that we can create one. Do we need one act that would actually encapsulate everything that is to do with genetically modified organisms, or how we actually going to use these technologies? Or are you happy that it will be embedded in different acts? And yeah, I would love to pose the question to all three of you.
I think I'm going to throw this one to Emily.
Yeah, I was going to say.
Would love to see it simplified. I just don't know how you simplify. This is a complex starting point where we are. It's a little bit like biotech. We're not a sector, we're a platform that sits across everything. And therefore it can't just be in one place, it sits in multiple. But handing over, can we have one act for everything? I don't know.
Well, not one act for everything, sorry, Zara. Life can't be that simple, unfortunately. And you know, that diagram you showed, Zara, is a huge issue about overlapping regulation and overlapping legislation as well. What is proposed is a single gene technology act. So all regulations associated with gene technology are dealt with via that act rather than via other acts, and to be really tight on purpose of that act. So it's really managing the risks only of gene technology to human health and the environment. And so that tightness on purpose will help with some of the different complex landscape for different pieces of legislation. You know, whereas the Animal Welfare Act will have a different purpose. So it's making sure the regulator is regulating the bit it needs to regulate, whereas other forms of legislation are managing other risks or other animal welfare issues or veterinary medicines issues or other things that are associated with that medical safety. And this act won't be saying that a technology or product is safe for human use. It's safe for human use. It'd be managing the, you know, safe for a medical application, for instance. That will be determined by the people with the appropriate skills. It will say that the risks of the gene technology are considered appropriately managed for work to proceed. And that's, as Sarah said, that's all about that nuance of being able to manage those risks appropriately to protect human health and the environment.
Fantastic, absolutely clear. Richard, do you want to comment, or you?
No, Emily and Zara have nailed it. Thank you.
Thank you so much, and this was absolutely amazing. And I think you brought a lot of knowledge and nuance to the topic, and I can't wait to what the future actually brings us. Thank you everybody online, and again, for the people that couldn't log on, I hope you enjoyed this recording. This is it from me. Thank you so much, and we will see you next time.